this video, we're going to look at question number 21 from GATE Biotechnology 2014 paper. So the question is, reaction between antibodies and antigens that are detected by precipitate formation in an agar gel are referred as option A, immunoprecipitation assay, option B, immunodiffusion assay, option C, immunoaggregation assay, and option D, immunofixation assay. So the answer to this question is immunodiffusion assay, and let us look more into the details of this technique and the other assays in this video. So immunodiffusion is a diagnostic technique for the detection or measurement of antibodies and antigens by their precipitation, which involves diffusion through a substance such as agar or agarose gel. So the advantages of this assay is that the reaction formed from this method is stable and can be preserved for staining and visualization later. It can also be used to detect identity, non-identity, and cross-reaction between antigens in a mixture, which we will look further in detail. So there are several types of immunodiffusion which are classified on the basis of the number of reactants that diffuse and the direction in which the diffusion happens. So the common types are single diffusion in one dimension, which is also called the Odin procedure, and double diffusion in one dimension, also called the Oakley-Fulthrop procedure. And there's also single diffusion in two dimensions, also called radial immunodiffusion or Mancini method. And then there's double diffusion in two dimensions, also called Octaloni double immunodiffusion. Let's first look at single diffusion in one dimension, which is also called Odin test. So the antiserum, which is the antibody, is incorporated in melted agar gel, and the mixture is poured into a test tube and it's allowed to solidify. So the antigen solution, as shown here, is placed above the agar. So the precipitation band appears in the agar like you can see here. So the antigen just diffuses through the agar gel, and as it reacts with the antibody in the agar gel, it forms a precipitation band. So then there's double diffusion in one dimension, also called the Oakley-Fulthrop test. So here the antibody or antiserum, like similar to single diffusion, is incorporated in agar and poured into the tube, and it's allowed to solidify. And then a second layer of agar without any antibody is placed above this and it's allowed to solidify. And then the antigen solution is placed above the agar. So now the precipitant band appears in the plain agar column because the diffusion happens in both directions. Like the antigens on top diffuse into the second layer, and the antibodies in the third layer also diffuse in the second layer, and as they react, they form the precipitation band. And then there's radial immunodiffusion, also called the Mancini technique. So the single radial immunodiffusion is used extensively, widely, for the quantitative estimation of antigen. That is, it's used to quantify how much antigen is there in a given sample. So the principle behind this assay is that the antigen and antibody reaction is made more sensitive by the addition of antiserum into the agarose gel, and then the antigen sample is loaded in the well. See, as the antigen in the well diffuses into the agarose radially in all the directions, its concentration continuously falls until the equivalence point is reached. So at this point, the antigen concentration is in equal proportion to the antibody concentration in the agarose gel. So at this point, the ring of precipitation, that is the precipitant ring, is formed around the well. So the diameter of the precipitant ring is proportional to the concentration of the antigen. For example, if there's higher concentration of antigen, the precipitant rings are formed with larger diameter. That is, it takes longer time and longer distance for the antigen to reach and reach the same concentration as the antibody. So that's why it has a larger diameter. Also, the size of that precipitant ring depends on the antigen concentration in the sample well and also the antibody concentration in the agarose gel, also the size of the sample well and the volume of the sample used. So thus, by having several concentrations of a standard antigen, standard curve can be obtained from which we can determine the amount of antigen in an unknown sample. So this is a quantitative test, and if we see more than one ring in this test, it suggests that more than one antigen or antibody reaction may have occurred. So this could happen when there's a mixture of antigens or antibodies. So this test is commonly used in clinical labs for determining immunoglobulin levels in patient samples. So the procedure is as follows. So an agar containing an approximate antiserum antibody is poured in plates. So circular wells are cut and removed from the plates. And then a single or a series of standards containing known concentration of antigens are placed in separate wells, while the control and the unknown samples are placed in the other remaining wells. So as the antigen diffuses radially, a ring of precipitate will form in the area of optimal antigen antibody concentration. So the ring diameters are measured and noted. A standard curve is prepared using the ring diameters of the standards versus their concentrations, and this curve is then used to determine the concentration of the control and the unknown sample. So how do we interpret the radial immunodiffusion? So the presence of a precipitin ring around the antigen wells indicate specific antigen antibody interaction, like we just saw. So absence of precipitin ring suggests that there's no antigen, or there's no reaction between the certain antigen and the antibody. So the greater the amount of antigen in the well, the farther the ring will be from the well. So this is how it looks like. So the red spots here are the antibodies. No, the red spots are the antigens that are in the well, which are diffused into the agar gel. And here you could see the antibody structures that are incorporated in the agar. And when the concentrations of antigen and antibody are similar, they form this precipitation ring. So if you look at a real time setup, this is how it looks like. So this is your Petri plate, and the circular cutouts of agar are the wells, and these are the precipitin rings formed. So this is probably the control where there's no antigen, that's why there's no ring formed around it. And so these here you could see the precipitin rings. So here you could see a ring very close to the well. So this means that there's very little concentration of antigen. And here you could see that the precipitin ring is very far from the well, which suggests that higher concentration of antigen is present in this well. So that's how you interpret radial immunodiffusion. So the applications of radial immunodiffusion. So immunodiffusion techniques are mostly used in immunology to determine the quantity or the concentration of antigens in a sample, and the estimation of immunoglobulin classes in serum, and also to develop relative concentrations of antibodies in serum and to compare properties of two different antigens. Let's now look at Octaloni double diffusion, which is also a type of radial immunodiffusion. So here the antigen solution is placed in wells in the gel plates, while the antibodies are placed in other remaining wells. So upon incubation, both the antigens in the solution and the antibodies each diffuse out of their respective wells. And in case of the antibodies recognizing the antigens, they interact together to form visible immune complexes which precipitate in the gel to give a thin white lining indicating a reaction has occurred. So in case multiple wells are filled with different antigens and antibodies, the precipitate developed between two specific wells indicate the corresponding pair of antigen-antibody reaction. So this test is performed by cutting wells like these in the agar gel poured on a Petri plate. So the antibodies is placed in a center well, and different antigens are added in the wells surrounding the center well. And after incubating these, in a moisture chamber, the lines of precipitants are formed at the site of the combination of antigen and antibody, like you can see here. So, the results of this method can be interpreted by either of these three ways. So, one is a full identity, that is a continuous line, as you could see here in this first image. So, the line of precipitation at the junction forms an arc which represents serologic identity or the presence of common epitope in antigen. So, like we said, the middle well over here is the antibody, and the other wells are antigens. For example, here let's say we have two wells where we have two two samples of antigens, and if we see a line like this, a continuous line like this, it either means that both the samples have the same antigen, or both the antigens have a common epitope. So they react with the antibody in a similar manner, and so a continuous ring is formed. And then, in case of non identity, two lines cross completely. So that is a pattern like this. A pattern of crossed lines demonstrate two separate reactions and indicates that the compared antigens are unrelated and share no common epitopes. So this suggests that like both these wells, they have completely different antigens and the reactions happen in different ways, and so like totally two different precipitation lines are formed. And then, in case of partial identity or a continuous line with a spur at one end. So, here the two antigens share a common epitope, but some antibody molecules are not captured by the antigen and traverse through the initial precipitin line to combine with additional epitopes found in the more complex antigen. So, the pattern of the lines that form can determine whether the antigens are the same or not. So, this technique is useful for the analysis of antigens and antibodies, and it's used to detect uh, and identify and quantify the antibodies and antigens such as immunoglobulins and extractable nuclear antigens. Also, agar gel immunodiffusions are used as serological tests that historically have been reported to identify antibodies to various pathogenic organisms. And the demonstration of antibodies in serodiagnosis of smallpox and also in the identification of fungal antigens. So that's it about immunodiffusion assays, which uh, is characterized by the formation of a precipitation line as a result of antigen antibody interaction. And then the one of the other options was immunoprecipitation assay. So what is this? Although the name suggests that precipitation is formed, that this is not the answer to our question. So immunoprecipitation is the technique of precipitating a protein antigen out of a solution using antibody that specifically binds to that particular protein. So this process can be used to isolate and concentrate a particular protein from sample containing many different proteins. Also, immunoprecipitation requires that the antibody be coupled to a solid substrate at some point of the procedure. For example, like a bead over here. So the antibody is attached to the bead, and the antigens isolated by immunoprecipitation are analyzed further by SDS beige or Western blotting. So, in short, this technique is used to separate out or precipitate out a particular antigen or a protein of interest using an antibody that is specific to it. The antibodies are embedded in beads, and these antibodies are used to extract these antigens of interest. And then let's learn about immunoaggregation assays. So, the rapid, sensitive, and low cost detection of macromolecular biomarkers is critical in clinical diagnosis, environmental monitoring, in research, and so on. So, these macromolecular biomarkers could be anything like nucleic acids, proteins, enzymes, hormones, etc. So, immunoaggregation assays are mostly microparticle based and is a method for the detection and quantification of macromolecular biomarkers. So, in general, antibodies are conjugated to microparticles through streptavidin biotin interaction. So the antibodies are biotin-related, and like microparticles like beads are coated with streptavidin, and the biotin in the antibody and the streptavidin on the bead, they interact to form conjugates, and these conjugates are detected by aggregates. So, the addition of macromolecular biomarkers cause the aggregation of these antibody microparticles in different size distributions, which are subsequently detected by an optical microscope or optical particle sizer or through any other detection methods. For example, in the absence of an analyte, that is the macromolecular biomarker, these biotin-related antibodies and streptavidin coated beads, they form big macroparticle aggregates. While let's consider the presence of an analyte like VEGF, which is a protein involved in angiogenesis, that is blood vessel formation, and it's detected mostly in cancers. So, in this case, VEGF binds to the biotin-related antibody, and the microparticle aggregates are formed only between the unbound antibodies and streptavidin beads, and hence small aggregates are detected. Now, based on the size of these aggregates, it could be interpreted whether the analyte is present or not. Its relative concentration could also be measured. Like here, like we said, small aggregates means there's high VEGF, and larger aggregates, it means either there's low VEGF or there's no VEGF, there's no analyte. So this is the basis of immunoaggregation assays. And the next one is immunofixation assay. So, immunofixation electrophoresis is a technique for the identification of proteins within complex mixtures after separation by either conventional electrophoresis or by isoelectric focusing. So, most commonly, antigens, which are often immunoglobulins, are separated by electrophoresis followed by precipitation with specific antibodies. So, the process involves the use of specific antibodies to detect and visualize different classes of immunoglobulins and their respective light chain types, the kappa and the lambda light chain types. So, the primary antibodies are directed against the different classes of immunoglobulins, which are IgG, IgA, and IgM, and the light chain types kappa and lambda. So this antibodies apply to the gel or the membrane, and then the antibodies selectively bind to specific immunoglobulins present in a patient's sample. So, and when they interact, when the immunoglobulin and the antibody against it interact, they react and can be visualized by staining. So, immunofixation electrophoresis is especially useful for identifying abnormal proteins such as monoclonal immunoglobulins, that is IgM, in conditions like multiple myeloma. So, immunoglobulins are generally proteins that function as antibodies and they fight infection. So, there are, like we know, there are these three types of immunoglobulins that fight different types of infections. But some immunoglobulins can be abnormal and maybe due to cancer and other conditions like multiple myeloma, like we just saw. So, in short, this assay is used to find the predominant immunoglobulin in a mixture of proteins in the patient's serum by treating it separately with antibodies directed against them. So, a normal result includes a darker immunoglobulin G, IgG lane, and a lighter immunoglobulin A and M. No, and a lighter immunoglobulin A and an absent immunoglobulin M. And usually the kappa is denser than lambda, but yeah, you might find these two chains as well. So, in case of an abnormal condition, in case of like multiple myeloma, like where there's abnormal IgM, you would find lighter IgG, absent IgA, and darker IgM. So, uh, through this, you can identify the abnormal immunoglobulins. So, that is the principle behind this assay. Now, coming back to our question, the reaction between antibodies and antigens that are detected by precipitate formation in an agar gel is referred to as. We already saw that the answer is immunodiffusion assay, where we can see visible precipitate rings. That is the answer. We also saw about the principles of the other three assays. Thank you.